The third component of the membrane which we are going to study is the membrane proteins. Membrane proteins. Now, many proteins in our body are actually found on the cell membrane. Okay, we have got two types of proteins in the cell membrane. The first one is called the peripheral proteins. Peripheral proteins, like the name suggests, they tend to be on the surface of the membrane. On the surface of the membrane, like this one and this one. Here, this is a peripheral protein. These are peripheral proteins. They are not going to go through the membrane. They are not embedded in the membrane, but they are stuck to the surface or bound to the surface of the cell membrane. However, on the other hand, we have the integral proteins. Integral proteins, as you see here, they go through the plasma membrane. This is an integral protein. And this is an integral protein. You can see the full protein, the complete protein is fully inserted in the plasma membrane. That's why we call it integral proteins. Integral proteins. Okay, so they penetrate the hydrophobic core of the lipid bilayer, and we also call them transmembrane proteins. Trans means they are fully inserted in the cell membrane. Now, let me ask you a question here. Let me pause and ask you a question. What do you think is the nature of the peripheral protein? What's the nature of this protein? Is it hydrophobic or is it hydrophilic? This one here, for example, do you think is hydrophobic or hydrophilic, and why? Why do you think so? Okay, the answer is, as you have guessed, it is hydrophilic because it's facing the cytoplasm. Okay, I'll pause a bit لأنه أذان العصر وبكمل بعد شوية. Okay, what about the nature of the integral proteins? What about the nature of the integral proteins? Do you think they are hydrophilic or hydrophobic? For example, this integral protein, is it hydrophilic or hydrophobic? It's penetrating through the whole of the plasma membrane. So as you might have guessed, this part here is going to be what? Hydrophobic. Hydrophobic. And this part here, let me select a different color. This part here is going to be hydrophilic. It's facing the external environment, which is made up of water, and facing the internal environment, which is also made up of water. So basically, the integral protein is no, it's not hydro, it's amphipathic, which means it is hydrophilic and hydrophobic. Right? All right, so now you know the structure of membrane protein. What is their importance? What is the function of membrane proteins? Let's look here. The first function is transport. They function in transport of molecules inside and out of the cell. So if you look at this figure here, you have a membrane protein that has a channel. You can see this channel. This is called the channel protein. Okay, and you can see things are being transported from outside of the cell to the inside. And you have got another kind of membrane protein here, the kind that changes shape. So you can see it opens from the inside, and then some molecules, they bind to the binding site here, and then it opens to the outside and the molecules are exported. Now, we are going to talk about these proteins in details in the next lecture, inshallah, when we talk about transport of molecules. All right, the second function of membrane proteins is enzyme activity. So these proteins that are located in the plasma membrane, they can speed up chemical reactions. So basically, they act as catalysts, biological catalysts. They speed chemical reactions. Just a normal enzyme activity. So basically you can see this is a substrate that binds to an enzyme and gives a product. This is a substrate, this is a product, and now this product is acting as a substrate to this second enzyme, and you have it converted to another product here. Okay, so this also acts then as a substrate. Okay, so that's the second function of membrane proteins. The third function of membrane proteins is cell signaling. Okay, they play a role in cell signaling. Remember when we talked about hormones and receptors in Unit 2.4? No, I think Unit 2.5 in the proteins lecture. You remember that? So this is a receptor. This is a membrane protein that acts as a receptor, and this is a signal, or you can imagine this as a hormone, okay, that binds to the receptor. The receptor binds, the hormone binds to the receptor, and it actually transmits a message, in this case from outside to the inside of the cell. The fourth function of membrane proteins is cell-to-cell recognition. And we actually talked about this point in details when we talked about carbohydrates, okay? Because whenever we talk about recognition and how the cell is able to recognize molecules, recognize self from non-self, then we always talk about carbohydrates. In this case, this carbohydrate is a glycoprotein because it's bound to the protein. Okay, so this is really important when you're fighting pathogens like bacteria and viruses, as we mentioned previously. And we call these things identification tags. Okay, identification tags. So it's like a tag, like a sticker on a cell that tells the body whether this is like a cell from the same body or it's a foreign body, etc., etc. The fifth function of membrane proteins is intercellular joining, which means joining or attachment between one cell and another. It basically forms as a hook. It acts as a hook, okay, that joins two cells together. We are going to talk about this later when we talk about gap or tight junctions. Remember, this is the last topic of our lecture today. We're going to talk about junctions in details. The last function of membrane protein is attachment. Basically, if you recall from last lecture, lecture 3.3, when we talked about the cytoskeleton, the proteins in the cell membrane, they act as a link between the interior of the cell. You can see the cytoskeleton here, okay, and the exterior of the cell, which is the extracellular matrix. This is going to be our next topic, inshallah, during this lecture. This is the ECM. So basically you see that this is, what is this? Is it an integral protein or it's a peripheral protein? Yeah, this is an integral protein. And this here, this is a peripheral protein. So you can see that the protein here acts as a link between the cytoskeleton and the extracellular matrix. All right, so these are the 6 functions of membrane proteins. All right, now that we have talked about membrane lipids, membrane carbohydrates, membrane proteins, let me ask you a question. Where are all these components of the plasma membrane synthesized? Where are they made? Let's look at this figure here. All right, so yes, as you can see, they are synthesized via the endomembrane system. Okay, let's start with number 1 here. You can see that the endoplasmic reticulum produces the transmembrane glycolipids and the transmembrane glycoproteins. So it's synthesized in the ER. So number 1, synthesis in the endoplasmic reticulum. Okay, so what's the next destination? The next destination is the Golgi apparatus. As you see in the figure, you can see that the transmembrane glycoproteins are transported to the Golgi apparatus, which does what? As we have studied in Unit 3.2, it modifies and packages transmembrane glycoproteins and glycolipids as well. So some modifications are going to take place as it moves from one cisterna to the next. So that's number 2. Next, vesicles are going to bud from the Golgi apparatus, and these vesicles, they contain the products of membranes or the membrane components, and they are going to be transported to the plasma membrane. So number 3, vesicles. This is number 3. It's done. 2 and 1. And we have number 4 left. Number 4, the vesicles are going to fuse. They are going to fuse with the membrane in order to release the components of the cell. So here you can see the glycoprotein. You can see the glycoprotein, which is the carbohydrate that is bounded to the protein component. So the carbohydrate, of course, it's the green thing here, and you can see it's bounded to this one here. Let me just omit this. Okay, this is the glycoprotein, and you can see also the glycolipid, where the carbohydrate is actually attached to the lipid part of the membrane. Okay, so these components are produced by the endomembrane system. They start in the ER, then to the Golgi apparatus, and then a vesicle buds from the Golgi apparatus, fuses with the plasma membrane, and you can see them here facing the exterior of the plasma membrane. So number 4 here would be plasma membrane. And this is how the components of the plasma membrane are synthesized. All right, so the title of this slide says that membranes have distinctive inside and outside faces. Okay, so the inside and the outside faces of the membrane are different. They are not the same. They are different. They are different. Okay, or we call it asymmetrical. 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 Okay, which means it's not symmetrical, which means it's not the same. Okay, the outside face of the membrane, this is the outside face of the membrane, is always different than the inside face of the membrane. It's always different. Okay, let's look at the differences. What are the differences between the outside face of the membrane and the inside face of the membrane? Okay, the first thing, which we already discussed previously, are the carbohydrates. You can see the carbohydrates are restricted to the exterior face of the membrane. They are always facing to the outside of the cell. Okay, and this is related to their function because their function is cell-to-cell recognition. Remember, these guys, they act as tags, so they have to be on the outside of the membrane, on the outside face of the membrane. The two layers also differ in the other components, okay? They differ in the integral proteins, they differ in the lipid composition. So all of these things, they differ. The carbohydrate, the lipid ratio or the lipid component, and also the integral proteins. The integral proteins. You can see here the integral proteins. They are found on the inside face. They can also be found on the external face of the membrane, but in this figure specifically, we see them more on the interior face of the membrane. All right, and this is the meaning of distinctive or asymmetrical faces or of the membrane. All right, ladies and gentlemen, now we are finally done with the cell membrane. Okay, we talked about the cell membrane in details. And now we are ready to go outside the cell membrane. Okay, we are ready to go to the surfaces of the cell. So we are going to start with the plant cells. Plant cells are covered or encased. Encased means covered by the cell wall. طبعا this one we took it in the very beginning in unit 1. We already mentioned that plant cells have a cell wall. Okay, so what is the cell wall? The cell wall is a protective layer. It provides protection for the cell and it's present on the exterior of the cell membrane or the plasma membrane. Okay, now can you answer this question? Which other cells other than plant cells, which cells other than plant cells also have the cell wall? I mentioned this in the first slide, and we said that bacteria has cell wall and fungi also has cell wall, but the components or the structure of the cell wall is different in each of these 3 organisms. Okay, let's look at the details of plant cell wall. Okay, so this is the cell wall of plant. Okay, so let me draw the plasma membrane of plant with this red color, and I'm going to draw cell wall in a different color. This is the cell wall. It's present on the exterior of the plasma membrane. Now, of course, you have the nucleus somewhere here. Just dotting it because you can't shade scientific drawing. All right, so now let's zoom into this cell wall. If we zoom in here between the two cells, we are going to see the cellulose microfibrils. These are like wires, okay, thick wires. Microfibril. This is a microfibril. Okay, now if we zoom in, you can see you have many microfibrils here. Now, if we zoom in to one of these microfibrils, we are going to get very thin wires here. You can see these very, very tiny hairs. They're very thin, unlike the microfibril, which is made up of many, many cellulose molecules. So these are cellulose molecules. Remember when we studied about cellulose molecules and we mentioned that they play a role in the structure. They are structural polysaccharides. Okay. Now if we zoom into these cellulose molecules, we see that the building block of the cellulose molecule is what? Yes, it's the beta glucose monomer. Remember beta glucose and alpha glucose? Okay, please link this to the unit of carbohydrates and refresh your memory about the difference between beta glucose and the alpha glucose. Okay, so basically here we went from the bigger picture to the tiny one. Now, if we do the opposite, the building block is the beta glucose monomer. You've got the bond here between one glucose monomer and the other one. What do we call this bond? Can you recall? Yes, it is called a glycosidic bond. Okay, it's a type of covalent bond. So they form the cellulose molecule, and many cellulose molecules together make up the microfibril, and many microfibrils together gives you the cell wall of the plant cells. Okay, so they are made up of cellulose microfibrils, and these cellulose microfibrils are actually embedded in a matrix of other polysaccharides and proteins. Okay, so there is a background of other polysaccharides and proteins, not just cellulose microfibrils. All right, so this is the structure of a plant cell wall. Okay, now let's move ahead with the functions of the cell wall. We already mentioned some of these functions while we were studying the plant cells in the lab. If you recall the Rio discolor, we observed the Rio discolor under the microscope. Okay, we actually extracted a layer or a tissue layer of the plant and we observed it under the microscope. You even measured its size under the microscope. So there are three functions of the plant cell wall. Number one is protection, of course, since it is present on the outside of the cell, so it plays a role in protection of the plant cell. And number two, it maintains the plant cell shape. Now tell me, how would you describe the shape of a plant cell? We already mentioned this several times in the lecture. Plant cells, they have a regular shape. They have a regular shape, unlike animal cells, which have an irregular shape. Why do plant cells have regular shape? It's because they have the cell wall. It's because they have the cell wall. All right. And the third function of cell wall is it prevents excessive uptake of water. Okay, so water cannot enter excessively into the plant cell. Of course, it can enter. Okay, water can enter, but there is a limit to how much water can enter the plant cell. Okay, and again, we are going to study this in detail in the next lecture, inshallah, and you're gonna see a difference in plants between plant cells and animal cells when water enters into the cell. How does each one behaves? All right, now let's look at the structure of the cell wall. The structure of the cell wall. Of course, we mentioned in the previous slide, we talked a bit about structure, but now we are looking at the bigger picture here, okay? All right, so let me draw another plant cell here on the top. Just a minute, okay, erase it. All right, so I'm gonna, because it's easier to understand when I make a drawing here, I'm gonna make a huge drawing. This is a plant cell with its regular shape, and now I'm gonna draw the, طبعا this is the plasma membrane, okay, the thing that I have drawn in red, and this is the primary cell wall. This is the primary cell wall, okay? So this is primary cell wall. Let me label this as plasma membrane. Okay, so here in this figure, this is the plasma membrane, and let me select a green pen. This is the primary cell wall. Okay, now the primary cell wall is flexible. It's a flexible cell wall that a plant produces at the very beginning when it's young. When it's young, it produces a primary cell wall. And then as the plant grows older, it starts adding layers of secondary cell wall between the plasma membrane and the primary cell wall. This blue thing here, this is the secondary cell wall. This is seen in older plants, okay? When they grow older, they produce secondary cell wall, and it is produced in layers. So this is a second layer of secondary cell wall, and this is a third layer of secondary cell wall. As it grows older, it produces layers, other layers, like additional layers of a secondary cell wall. Okay, so that's secondary cell wall. Here you can see that it's made up of layers. Can you see these lines here? Okay, so it's made up of layers of secondary cell wall. Unlike the primary cell wall, the primary cell wall is only one layer and it's thin, okay, and it's flexible, okay. Now, if we look into the connection or the space between one plant cell and the next plant cell, you've got here in the middle what we call the middle lamella. The middle lamella here. Now, this middle lamella is a sticky polysaccharide that sticks two plant cells together. Okay, it sticks two plant cells together. So this is known as the middle lamella. Now, you should keep in mind that not all plants have a secondary cell wall. Some of them, they just have primary cell wall, okay? But all plants must have a primary cell wall. Primary cell wall. Okay, now, do you know that from the secondary cell wall, we can get information about the age of the age of plants, okay? So, you know the layers of plant, when you cut the trunk of the plant, you get these circles here. This can give us an idea about the age of the plant, okay? So basically, the secondary cell wall, unlike the primary cell wall, this is tough. This is tough, unlike the primary cell wall, which is thin and flexible. All right? Okay. All right. As we agreed previously, animal cells, they do not have cell wall, but they have another structure that is found outside the plasma membrane. This structure is known as the extracellular matrix, or the ECM. The ECM. The ECM is composed of a number of components. You can see in this figure, we have the collagen fiber. We have the collagen fiber over here, and we have this matrix of proteoglycan complex, and we have here the fibronectin. This is the fibronectin. And finally, we have integrins, which are these transmembrane proteins. Okay, so the main component is the collagen fiber. You can see here they're huge. I'm sure all of you have heard about collagen. Collagen is found under the skin, and it gives the skin its flexibility. As we age, as we grow older, we have less collagen, and this makes the lines appear, or the wrinkles. Okay, and proteoglycan, this is the whole complex here. We have fibronectins, which join between the collagen fiber and the integrin. And we have integrin, which is a kind of protein. Okay, it's an integral protein. An integral protein, you can see it's embedded in the membrane. All right. So the main component, as I have mentioned, is the collagen fiber. This is the main component of the extracellular matrix. It's a fibrous structural protein. Do you recall we already talked about collagen in the protein lecture, and we mentioned that it's a structural protein. Then we have the fibronectin and the integrin. These are the adhesive protein because they join, especially the integrin. It joins, it acts as a link between the inside of the cell and the outside of the cell. Integrin. Okay, and then we have this proteoglycan complex, which looks like a tiny root or tiny leaves that bud here in the matrix. Now, if we look at this figure here, you can see this green line. Let me choose the green color. This green line here, this is a polysaccharide molecule. And then coming out from this polysaccharide molecule, you can see this purple lines here. Okay, if we zoom in this purple line, this is a protein here. That's a protein. And coming out or budding from this protein, you've got these carbohydrates. So this is called a proteoglycan because you have protein with carbohydrate. So we call it proteoglycan. And you've got this polysaccharide, long polysaccharide molecule. So from the name, you know that this is protein and carbohydrate. Okay, protein and carbohydrate. And that's about the extracellular matrix. All right, so we have seen the structure of the extracellular matrix. Now, let's talk about its functions. The functions of the extracellular matrix. Number one, the first function of the extracellular matrix is mechanical support. It gives the cell mechanical support. Okay, let me choose a highlighter. That's number one. It gives mechanical support. It acts as an anchor for the cell, just like the function of the cell wall in plant cell, which provides support for plant cells, right? So for animal cells, we have the extracellular matrix, which provides support for the animal cells. The second function is adhesion to surfaces because it has these adhesive molecules. The matrix itself, the matrix of proteoglycan, it's located on the outside, so it helps the cell stick to surfaces. So adhesion to surfaces. Okay, and the third function of the extracellular matrix is movement of chemical molecules and food particles. So it helps in the movement of these particles. Since it's present in the outside, that's what we expect. Finally, one of the most important functions of ECM is regulation. Okay, regulation. It regulates the changes in the gene expression. How does it do that? I mean, the genes are present inside the nucleus and the extracellular matrix is on the outside. How can something on the outside control something which is really in the core of the cell? It's not in the cytoplasm even, it's in the nucleus, which is protected in the nucleus. How does it do that? Let's look at the next slide over here. Let me erase this. All right. So as you see, we have the extracellular matrix. You can see there, there is a mechanical signal. Mechanical means it's touching. They are touching each other, okay? This is the ECM. So you have fibronectin that is bound to the integrin. Remember, integrin is in the plasma membrane, so it's actually the link. This is the link between outside and inside, okay? So fibronectin to the integrin, and then the integrin is bound to the cytoskeleton. Okay, these are all bound together. That's why we say it's mechanical, because they are linked, mechanically linked together. But then from the cytoskeleton to the nucleus, it's not bounded together, but there are chemicals. That's why we call it chemical signaling. So chemicals are transferred until they get into the nucleus, and when they get into the nucleus, they will affect gene expression. Okay, so this is our double helix, and it will affect gene expression, and this is how changes in the external environment are going to lead to changes in gene expression. It's all because of this link here between the outside and the inside. Okay, so we have the cellular communication. All right. All right, so we have learned about the cell membrane. We have learned about the surfaces. In case of plant cells, it was the cell wall, and in case of animal cells, it was the extracellular matrix. So now we're going to go forward and study about intercellular junctions, which are the junctions between cells or neighboring cells, okay? The connections, التقاطع ما بين الخلايا, okay? Intercellular junctions. So animals, they have three junctions. Have the tight junction, you can see it here, and they have desmosome, and they have got the gap junctions. Okay, 1, 2, 3. On the other hand, plant cells, they have just one intercellular junctions, and this is called plasmodesmata. Plasmodesmata. Plasmodesmata is found in plants. You can see plasmodesmata here. These are tiny channels, okay? Plasmodesmata. All right, so in the next few slides, we are going to learn about the structure of these junctions and the function of these junctions as well. So all of these intercellular junctions are found in epithelial tissues. They are found in epithelial tissues. Can you recall epithelial tissues? Have you seen this word before? Yes, indeed, you have seen it before. Remember the cheek cells? Remember when you observed your own cells under the microscope? And it was very interesting. You took epithelial cells from the cheek, from the inside of your cheek. These are called epithelial cells. So epithelial cells are a specialized group of cells that line the internal surface of the body. It lines the outer surfaces of organs, blood vessels, and even in the inner cavities of many internal organs like the stomach, the kidneys, the intestine. Okay, so epithelial cells, they line the internal surface of the body. Okay, so let's start with the first intercellular junction. This is called the tight junction. The tight junction. So from the name, you can imagine this junction to be very tightly holding the membranes of neighboring cells together. So it's like they are pressed against each other. They are pressed against each other, and you can see kind of a tiny or a thin rope that holds the two cells tightly together. This is very, very important because it helps in preventing leakage. Okay, so it prevents leakage of what we call the extracellular fluid, which is the fluid present on the outside of the cell. It prevents the leakage across the layer of epithelial cells. So water or any liquid can never pass through the two cells. Okay, water can never pass here. So no H2O. Now, why is this important? Why is preventing the leakage of extracellular fluid important? Okay, let me give you an example. For example, the bladder which holds the urine, okay, it holds a large amount of urine. We do not want the urine to penetrate between the cells because it's toxic, right? So we don't want the urine to penetrate between the cells, okay? That's why we have a tight junction. Okay, now this was number 1. Number 2 is desmosomes. Desmosomes, they are also called anchoring junction. anchoring يعني مرسى. Okay, so you can imagine these are very, very tough. They are very strong and they are made up of keratin proteins. Keratin, do you remember keratin proteins? We studied this in the proteins lecture. We said keratin proteins makes part of our nails. It's the structural protein and our hair. So it's fastening the cell. You can see these keratin keratin proteins. Okay, so in desmosomes, water can still pass here. Its function is not to prevent leakage. Its function is to fasten the cell, to keep them held together very strongly. You can see how tough is this structure here, which is made up of keratin. Okay, it's made up of keratin. So the third kind of intercellular junction in animal cell is known as the gap junction. Gap junction is also known as the communicating junction. From the name, we can guess what is the function of the gap junction. So basically, let me draw two cells here. This is one cell, and this is another cell. Okay, let me select another color right here. These junctions, they act as tunnels, tiny tunnels that allow molecules, small molecules, to be exchanged between one cell and another cell. That's why they are known as communicating junction. Communicating junctions. Of course, these are membrane proteins. Okay, you can see this is a membrane protein that is closed, and it can also be open in order to allow molecules to pass by from one cell to the next cell. Okay, but molecules that pass or that can pass through this membrane proteins have to be small molecules. Okay, they can't exchange large molecules. Only small molecules can be exchanged, such as ions, sugars, amino acids, and other small molecules can pass through the membrane proteins of the gap junction. All right? Okay. Now we move to the plasmodesmata in plants. This is the only intercellular junction in plant cell. It's called plasmodesmata. Plasmodesmata. طبعا plasmodesmata is also a communicating junction, so it has similar function to the gap junction. These two, they have similar function, okay, but they have different namings. In animal cell, it's called the gap junction. In plant cell, it's called the plasmodesmata. So just look here. This is the plasmodesmata. You can see this is a tiny channel that can allow small molecules to be exchanged between one plant cell and its neighboring plant cell. Okay, so it connects the cytoplasm of two cells together. So again, ions can be exchanged, sugars can be exchanged, and amino acids can be exchanged. All right, so this takes us to the very end of this lecture. Of course, this is a fun fact, which means you are not going to be tested on this, but it's just general information. Let us see what it says here. Proteins called connexins form the building blocks of gap junctions. Okay, so this is connexin, this is the connexin. Connexin mutations, so if you have a mutation in connexin, it can lead to cataracts, which is a disease in the eye, and a hearing loss. Okay, so actually this slide gives you the importance, shows you the importance of the proteins that are involved in the gap junctions. If there is a mutation, so this protein is changed, then this could lead to a disease in the eye, or it can lead to a hearing loss. Okay, it depends where is this connection mutated. All right, so this takes us to the end of this lecture. I hope you had fun with me, and I hope that you learned a lot from this lecture. Please, if you have any questions regarding this lecture, let me know or send emails to your instructors, and we will get back to you as soon as we read your question, inshallah. See you in the next lecture, lecture 3.5. This will be the end of Unit 3, inshallah. Have a lovely day, and thank you for your attention.